those things may coincide. So enough experience on the clinical side plus the manufacturing side coming together and may allow us to treat many more common diseases, which could be really transformational. I mean, it really could be amazing to have one-time treatments that deal with problems so that people don't have to take pills on a daily basis.
Welcome to Science with a Twist, a podcast for curious people who enjoy exploring how science impacts our daily lives. From technology that helps the fight against COVID-19 to solutions that help clean the water we drink, it's all thanks to science. In each episode, members of Thermo Fisher's scientific team talk to experts who are on the cutting edge of redefining how we exist. Let's get started.
Hello, and welcome back to the Science with a Twist podcast, brought to you by Thermo Fisher Scientific, the world leader in serving science. My name is Dr. Timothy Miller, today's host and Vice President of Enterprise Science and Innovation at Thermo Fisher. As our CEO, Mark Casper, continually highlights, we are now in a golden age of biology, enabling the science to truly deliver profound advances to human health. And our early efforts in gene therapy are playing, I think, an integral role. I'm really looking forward to today's discussion with Dr. Peter Marks, who you may be familiar with through his work as the director of the Center for Biologics Evaluation and Research at the FDA. During today's episode, we'll dive into the progress being made in gene therapy and what's needed to get these groundbreaking treatments to more patients and faster. A brief overview of what we'd like to talk about today are gene therapies ushering in this new era of medicine. I think that's because of their potential not just to treat symptoms, but actually to address the root cause of many of these diseases. 2023 was a landmark year for gene therapy development. We saw approvals across some of the most difficult diseases historically to treat, and also with incredibly high unmet medical need. Very exciting to myself, someone who's dedicated most of their career in the rare disease area. One example is last year we made history with the first ever approval for a muscular dystrophy, specifically in Duchenne muscular dystrophy, that was approved in a group of patients aged 4 to 5. It's a common rare neuromuscular disease affecting about 1 in every 3,500 male newborn children. And working with a number of clinical centers across the country and the globe, we've seen this first therapy come into approval. I think it's a groundbreaking event in medicine and was preceded by other approvals for gene therapies in the recent year. And I think many more in the future. For today's episode, we're privileged to talk with Dr. Peter Marks, who, as I said, serves as the director for the Center for Biologics Evaluation and Research at the FDA. Dr. Marks joined the agency in 2012. He's authored more than 100 scientific papers and has been a member of the National Academy of Medicine since 2022. I would highlight a recent article within the past week or two, specifically addressing some of the issues that we're noting now with CAR T therapy and helping to drive that to a better place for patients and families. Most recently, Dr. Marks and his team launched START, an FDA pilot program focused on advancing new treatments for rare disease populations. And this program has been likened to Operation Warp Speed, which played a key role in the support and acceleration of COVID-19 vaccine development during the pandemic. Welcome, Dr. Marks. Thank you so much for joining us here today.
Thanks so much. It's really a pleasure to be here, given all of the excitement that's going on in this field of gene therapy at this point in time.
That's fantastic. I know you've been dedicated to this area in a number of ways. I wonder if in your role at the Center for Biologics Evaluation and Research, can you talk about what you do there and how that sets the tone for some of the progress we're seeing in gene therapy now?
Yeah, so the Center for Biologics Evaluation and Research regulates some of the more complex biologic products. We regulate blood products, vaccines, cell and gene therapies. And actually, as turns out, it's kind of nice to have that cross-fertilization because, as you noted, some of what we learned with vaccines is now rubbing off and helping us in the gene therapy world. So we look at products and actually do basically start off with product-related research that gets done in some of our labs. It's applied scientific research, so we actually get a sense of the products we regulate. We also then obviously make policies regarding the products that we regulate, issue guidance to help people understand how to move things through the development process. And obviously, we receive applications and oversee the investigation of new drug process and then the biologics licensing applications to make determinations that the products are safe and effective and get them to market. So it's the real span of things. And I actually just neglected to say, we also then, after they are on the market, continue to follow them in post-market surveillance. So we really have kind of a cradle-to-grave approach of these products from kind of conception out through their use.
It's very exciting to hear that you are involved in so many different areas there. I think for many people, gene therapy is kind of a complex and diverse category of therapeutics that maybe include gene replacement, gene silencing, gene editing, and other modifications. And I mean, I wonder if you might just have a sentence or two on how you view that aspect.
Right. So gene therapy is really a broad area. And right now, just to think of the big buckets, we have these cell-based gene therapies where one takes cells from the body, out of the body, modifies them by adding a gene, potentially, or genes, and then gives them back. That's one of the simpler, in some ways, cleaner ways of making genetic modifications, although it involves additional complexity with getting the cells out and putting them back in. And then we have directly administered gene therapies, which come in essentially at least two flavors. One is when the gene therapy is administered locally, such as into the eye, or the other flavor is when they're administered into a vein, so they're given systemically and then target to the organ of interest. So really those are the big buckets, and then there are sub-buckets of that because the gene therapy itself can do exactly what you said. It could just add back a gene or it could modify a gene, and now with the CRISPR-Cas9 technology, which is an incredibly promising technology that has the potential to do many different editing functions to DNA, such as silence things or potentially fix a single base pair or potentially add in a stretch of DNA. Could even may be able at some point to add in a whole missing gene. This CRISPR technology is really quite exciting. So we have a lot going on in this space with a lot of promise. And even though we're still kind of early on, I think a lot to look forward to over the coming years.
That's great. You actually answered the next question that I was going to lead into about, can you talk a little bit about the progress over this past year? I think we've all seen really incredible achievements in regard to approvals, clinical data that's being generated, and of course, the very exciting work on at least two gene editing technologies that have been able to come forward. Very exciting for patients. Do you expect this level of momentum and the momentum of approvals to continue to increase at this pace or even greater?
Yeah, well, I'm hoping we can. I mean, to me, to see a technology like CRISPR genome editing make it from essentially the first paper published to an approved product in just a little over 10 years, that means it's, we're talking about the first scientific description of the system in a paper to actually having a licensed product to give to patients. I mean, I think that's tremendously exciting, and I'm hoping that we can keep up this type of pace of progress. I think we're trying to do, and I think we can probably talk a little bit more about this, we're trying to use all the tools in our toolbox to help pick up the pace of progress here, particularly in rare disease gene therapy, because now we have within our grasp the ability to take care of so many single gene disorders and potentially other disorders as well. And it's a matter of figuring out the way to leverage efficiencies to be able to bring these benefits of gene therapy to more individuals on an ongoing basis and to have many more approved products to keep up this momentum.
Yeah, I think you actually touched on a point that I think we've probably talked about before, but a lot of the focus in these early years has been on rare diseases. I think one, because of the incredible unmet medical need. I think two, because many of them, not all, but many are single gene disorders, and usually even a subgroup that is single base pair mutations that we believe we can find a way to fix or address specifically by replacing a gene, turning something on or off. How do you think this sets up our future for dealing with more common diseases?
Oh, I think this is absolutely critical because I think ultimately what is going to be probably a coincidence. I think it's like, I almost feel like this is one of those calculus problems when two trains are approaching and they're going to meet somewhere in the middle. I think what's going to happen is our ability to understand the safety and application of these gene therapies and how to have a very safe application of gene therapy will get to the same place about the same time as our ability to make very large quantities of gene therapies to treat large populations. Because for those who aren't familiar with this, manufacturing gene therapy today is not a cakewalk, to put it mildly. It's a challenging thing to do because oftentimes they have to be manufactured using producer cell lines, and the yields are not great. The purification processes are challenging. So it's not like making a small molecule drug. It's making a much, much more complex product. But several years from now, I think we can start to see over the horizon that advances in the technology, potentially to getting to systems where one doesn't need this type of producer cell lines, when one might be able to use actually a fair amount of synthetic biology to get there. Those things may coincide. So enough experience on the clinical side plus the manufacturing side coming together and may allow us to treat many more common diseases, which could be really transformational. I mean, it really could be amazing to have one-time treatments that deal with problems so that people don't have to take pills on a daily basis and get better outcomes on top of that. So this is really, I think it's coming together. I think it actually is kind of very nice because we're getting our experience in a population where the benefit risk is incredibly in favor of trying these therapies, and where we'll get worked out any safety concerns, I think, so that we're ready to go in larger populations.
Yeah, I love it that you dive right into the hurdles, which I think are key. And it's something that me and my colleagues around the globe think about every day. You mentioned a few of them, some of the basic science things that we might be able to bring to play here, some of the advanced analytics, some of the work around using structure-based biology to define these drugs in a better way. And then, of course, the clinical end, using real-world evidence, using long-term digital and virtual applications, which is exciting. And of course, no discussion about this would be complete without talking about, I think, the burden of manufacturing and doing that in a safe and appropriate way. I mean, interestingly, I think that the agencies around the globe have continued to think about what type of release testing, what type of stringencies are needed to make certain that we have not only appropriate safety for these drugs, but also to look at areas that we might be able to enhance their effectiveness. So it's very exciting to hear you talk about that. Do you see a role already starting to emerge for AI and other digital applications within gene therapy per se? I just want to give you a chance to hit on that because I know it's something that we're talking about every day.
Yeah, so I mean, I think there are so many different places. It goes across the whole spectrum from product design, because I think we're going to see AI used, especially with CRISPR-Cas9, to help us determine off-target effects in a very sophisticated manner, to be able to go through many, many, many genomes to get you the best estimate of whether you're going to have off-target effects. So that's on product design. I think we'll see it in clinical development with AI helping us to identify safety signals that humans just can't do because they can't look at so many parameters and integrate laboratories and clinical findings quite the same way. And then I think also we're going to see it on the manufacturing end, where AI can look at the incredible quantities of data that may come from multi-parametric monitoring of a biologic manufacturing process and tweak things, between connecting up a quality attribute at the end to various monitoring parameters, by the temperature, just simple ones like temperature, pressure, etc., but more complex ones that you can do continuous sensing on. AI can take all that data and say, oh, do the yields get better or worse when the temperature goes up or down a small amount? And ultimately can develop loops that lead you to what's essentially continuous process improvement. So I think this is, to me, AI at its best. This is where AI is helping humans. And I think it's like one of the examples that I always bring out, that when people get scared of AI or don't like AI because they don't like AI making art or writing books, this is AI doing what I think it can do really well for humans, which is really help us in this situation in a way that humans can't look at a million data points from 10 different parameters that might come in one day yet let alone multiple days.
Dr. Marks, one other thought that seems interesting to me is we're starting to talk about some of the issues that we've seen, some of the challenges, including things like vector immunity. Of course, we've seen some challenges in the trials, but I think we're starting to think our way past some of those issues. Some of that might be moving away from those vectors or utilizing other entities. Would you have some thoughts there?
Yeah, no, thanks so much for that question. There are really some interesting things here. Now, people are trying to make better generations of our current vectors, those that use adeno-associated virus, that will be either less immunogenic or that can substitute for one another. But then there's the issue of, could we just move away from this entirely to vectors that could potentially be given multiple times if necessary, because they don't cause the same immunity. And so this is quite an exciting thought, and this may be, as we move into this kind of synthetic world of what could be the gene therapy that's carried in by CRISPR-Cas9 in terms of an mRNA or a protein mRNA complex encapsulated in a lipid particle, which may allow us to achieve the same thing as we're currently achieving with viruses. It's a little ways off, but actually not all that far off. So I think this is really exciting, and that goes to this kind of bridge to more robust manufacturing, because when one is going to manufacture, we can manufacture RNA and, for that matter, protein relatively robustly and then encapsulate it pretty robustly. We know that from what happened during the COVID-19 pandemic with the mRNA vaccines, which is probably something we'll be able to do much more facilely for very large populations than make adeno-associated viral vectors, where the limitation there is kind of the yield that one gets from the cell lines, which, yes, we might get modest improvements in cell yields, but I don't see us getting to that transformational place where we're going to go from being able to treat hundreds of people to hundreds of thousands of people the same way as I might with the synthetic piece. So we'll see. I could be wrong, but it's a great question looking forward.
Well, I think this type of mechanism of working in some smaller areas is really well adapted here. We've seen this in other areas of medicine. A great example is the work in vaccine therapeutics. And I know internally we've worked now that we can develop a full human vaccine from concept, from targeted sequence to manufacturing to clinical trials in one sequence. We think we can do that in a couple months, actually 3 or 4 months. We believe we can do that now. And we're going to see that play a role, certainly in that area. I think we're also seeing this in gene therapy. I love it that every conversation I hear about gene therapy is also focused on the manufacturing and the needs of that. And I do think that it's critical. I think for me, the other thing is bringing this back to patients. I've participated in some efforts to screen populations looking for vector immunity and had to actually have some of those conversations with patients and families that are at higher risk for those and maybe not eligible for those trials or therapeutics. So I am excited to see the work in lipid nanoparticles and other types of vector technologies come through, but it's exciting to hear you seeing so much positivity there also. One of the ways in which you and I have gotten to know each other a little more is through work that Thermo Fisher and many other companies are now supporting, the Bespoke Gene Therapy Consortium, which is sponsored by the foundation of the NIH and part of the Accelerating Medicines Partnership that is put together. We're doing work there that I think is really delivering value. I wonder if you might have some comments on what you think will be the critical values coming out of this public-private partnership.
Yeah, no, I have to thank Thermo Fisher, thank people, all the different components that are working on this, because this is really an effort to figure out, can we put together a playbook ranging from manufacturing, the vectors, on through manufacturing of the vectors, out to the clinical development, to help people understand how we can get better throughput through the gene therapy development process. The idea here isn't like to make, although there are 8 or so diseases in the process, might be a little more, might be a little bit less, but roughly 8 diseases in the selection process that have been chosen. It's not about just those 8. It's about getting the playbook right so that as we move forward, we understand kind of the best practices in manufacturing, the best practices in clinical development that can ultimately allow us to improve the throughput through the process, potentially getting towards things like platforms. Now, we didn't bring that up yet, but this concept is gene therapies are somewhat different from small molecule drugs in that small molecule drugs, each one has its own unique fingerprint. And it's true that some are similar to one another, but gene therapies for a given gene therapy vector type, and there are specific types, some target different tissues, different ways, but for that specific gene therapy type, each one of the gene therapies actually, because of the size of the vector that's carrying it, is more like its sister or brother than we often have with drugs, because it's the insert, the gene of interest, that's different. And so it may turn out that we find ways to really leverage being able to move from one gene therapy to another without having to reinvent all of the information that's necessary about the manufacturing process, the toxicology process, and it may really help us to do some regulatory streamlining, which is really necessary if you want to get through hundreds of rare diseases, because to go back and do the same toxicology, the same manufacturing information over and over again is expensive, time-consuming, and it really slows down the throughput for the process.
And all of that's costly too, right? Costly in terms
Right,
of dollars,
exactly,
and costly
yes.
and costly in terms of development time, which is always something that I think we're focused to. You know, my CEO, Mark Casper, says every day he's talking to customers and probably to all sorts of audiences that we've teed ourselves up to really serve science. And when I think about the way that the Bespoke Gene Therapy Consortium is trying to drive that information, that knowledge, and thinking about it across that value chain that includes the early scientific endeavors, the manufacturing, the clinical trials work, the long-term follow-up. And I'm sure someday we will continue to make major advancements with regard to accessibility and getting these opportunities for patients to have it. I mean, I think about this as a neurologist and a researcher and as a member of a pretty diverse therapeutic development community now. I continually think about both the opportunity and really the obligation that we have to patients and families. And I've heard you say this before. I wonder if I might ask you if there's something specific that inspires you most about the gene therapy realm of therapeutics and why you're so hopeful for the future.
Yeah, so I get particularly inspired in this area because I've interacted with enough patients and their families. And to me, each one of these different rare diseases is a story, oftentimes of a patient that's affected by a terrible disease, and then the effect that disease not only has on the patient, but on the whole family unit. And the ability to intervene and have something transformational, it's so beautiful because it doesn't just transform that individual's life, that individual child, but an entire family, and in some cases, an entire community. A great example of that is with Duchenne's. Now, gene therapy may not work in everyone and it may not be perfect, but right now, when you see children that were starting to decline, that get a gene therapy, that then stabilize and are now out a few years, still running around when they normally would have been starting to end up confined to wheelchairs, this is amazing. And for the parents, it's just transformational. For their extended family, the grandparents, the cousins, the friends, it's transformational. And it's evidence actually to their whole community of actually science at work, medicine at work, making a difference. So I think it really, if we do gene therapy right, and we have to do it carefully, make sure it's safe, make sure that it's properly effective, but if we do it right, it's a shining example of kind of medicine at its finest.
Yeah, and I love it that we're kind of rounding this out, talking about Duchenne. I don't know if I've told you before, but I've spent hundreds of hours in the clinic with families and patients with Duchenne. I've worked with several of the preeminent patient advocacy organizations, I think, and the work that they are doing alongside what's being done by a huge tranche of companies across the industry that are really focused to that problem, culminating in a gene therapy now that, as you said, has tremendous value, to some groups in that population, is incredibly exciting. And it's something that drove me into industry and continues to inspire me on a daily basis. So I really appreciate the time that you've taken today to talk about this. I think that we have many things in common across the industry and across this therapeutic development world that we can continue to drive for patients. And I hope that we'll have a chance to have further conversations in the future around this.
Thanks so much. I really appreciate it and really look forward to continued collaboration in this area.
Excellent. Well, thank you for joining us today, Dr. Marks. It's such an honor to have you here on the podcast and around such an important conversation.
Thanks for tuning in to this episode of Science with a Twist. This show is brought to you by Thermo Fisher Scientific, the world leader in serving science. If you enjoyed this episode, then follow Science with a Twist wherever you get your favorite podcasts.